Hello, everyone. I'm Arnaldo Carreira-Rosario. I'm a postdoc at Stanford, and we'll be moving to Duke University next summer. So we're going to deviate a little bit, or maybe take a U-turn to some of the topics that we touched on before. So I'm really fascinated by this widespread phenomenon, and that's how the nervous system becomes active before we really interact with the world. So this is an example here of a developing retina. This is in ferrets before the animal can actually see anything. The retina is active. You see this also in zebrafish. This is a developing spinal cord before the zebrafish is moving anywhere. The spinal cord is active. And we're all familiar with this, especially moms, how humans also move before they can go anywhere. And so the question here is what triggers this activity for the very first time, and how this activity shapes the function of the nervous system. I'm using the fruit fly as a system, and the big advantage here compared to other systems is that you can really pinpoint the very moment when the nervous system transitions into become active. So we're looking at a single Drosophila embryo, fruit fly embryo. That's when the nervous system becomes active. And here, you can see first there are these movements, but you don't see any neural activity within the CNS. These are myogenic movements, so the muscles contracting on their own. Then you will see how the nervous system becomes active, and this is spontaneous network activity. And this correlates with movement. So now the nervous system takes control of the muscles. So the nice thing about this system here is that I can image not only one, but many embryos in parallel. And I can image this from the very beginning all the way until the animal hatches. So you can see here, they don't mind being photographed for hours. So this is nice because you have a system that you can basically screen for neurons, genes, maybe molecules that could be important for this process. So using this, I have found that a specific sensory input that is quite universal that we use to sense our muscles seems to act as a brake on this. So if you remove those sensory inputs, onset starts earlier, and then you also end up with changing the behavior of the animal. So the animal spends more time crawling as opposed to pausing and bending, which is important for finding food. More recently, I found that neuropeptidergic signaling acts as an accelerator. So when you block the specific peptide, you end up delaying when this activity starts, and you have less intensity in these episodes. So moving on, we'll be starting my lab at Duke University in the biology department. It's actually a building where my lab will be set up. We're renovating the area now. And so the questions that I'm interested in following up in is how does this intrinsic muscle twitching that occurs before the nervous system becomes active influences this network activity. So we're normally thinking of nervous system is active, telling something to the muscles. In this case, it's the opposite. The muscles are telling something to the nervous system, which is quite fascinating. The other question is how can these circuits become spontaneously active for the very first time in a precise manner? So something that I didn't mention is that these all occur very stereotyped. So the nervous system becomes active always at the same time. What are the developmental mechanisms controlling that timing? And lastly, how is that this activity shapes the circuits that are underlying these behaviors? So thank you for your attention. And I guess there are also ways here to collaborate with, if you want to ever look at how pollution affects invertebrate development, we can also look at that. Thanks.